Chapter 11 is going to look at the prokaryotes, how we characterize them and how we classify them. Now, in terms of the diseases that they cause, we'll be looking at that later. So we're not going to focus on that as much, just more on the characteristics of them. Prokaryotes are a very diverse group. They will survive and thrive in various habitats. They can be in the air, they can be in the land, they can be in the water. Only a few of them actually will cause diseases in humans. Keep in mind in general that of all the known microorganisms, whether you're talking about prokaryotes, eukaryotes, viruses, etc., only about 5% of them are human pathogens. In terms of their morphology, there are lots of different shapes. Most people think of the coccus, which is a spherical shape, then bacillus, which is rod shape. Now, as with anything, you have some varieties of these. Sometimes some organisms, they're kind of in between. It's like, is that a circle or is it a rod that kind of got squished? And so we call that a coccibacillus. Vibrio is kind of a curved rod. Spirals, some organisms have kind of almost a helical shape to them, that they could be called spiralium or spirochetes. There are certain species that are classified as being pleomorphic. That means they don't have a defined shape. It changes. Some may be rods, some may look like cocci, some may be a little bit twisted, and some actually are more like a star shape. So there's quite a variety. That is something that should be noted when you are trying to identify is when you look at a gram stain or a simple stain, what's the morphology? What does it look like? Some organisms do produce endospores, and not all will, but some do. We do see the gram-positive bacteria in the genus Bacillus and in the genus Clostridium. Those both will produce endospores. The vegetative cell is the cell that's actively growing right now. It will produce the endospore when it hits unfavorable environmental conditions. So it is a survival mechanism. It's not for reproduction, it's survival. So each individual vegetative cell would produce only one endospore. Endospores are a problem or a concern, at least in terms of healthcare professionals in trying to sterilize things. Their endospore is very resistant to heat. It's very resistant to some of our standard practices like moist heat, dry heat. And it's one of the things that we will use to gauge how effective various drugs are. Did it kill the endospores or not? And so there are different diseases that can be caused by organisms that do produce endospores, so that's why you want to make sure you can kill them. It's a concern to the food microbiology area. Food processors want to make sure that their final food items are not contaminated with these endospores. Excuse me. To give you an example, Clostridium tetani, Clostridium botulinum are different species that do produce these spores, and the bacteria can cause such diseases as tetanus, botulism, gas gangrene. For Bacillus, one of the huge concerns and why it says governments is there is a species, Bacillus anthracis, that does produce an endospore. That is the causative agent of anthrax, so there's concern for that. Not only from a healthcare professional standpoint, but certainly in terms of governments and basically biological warfare. So this is showing in the slides, the vegetative cell is the cell that's actively growing, and then the spores you can see, sometimes they're in the middle of the cell, sometimes they're on the end of the cell, but they're inside the cell. In terms of reproduction with prokaryotic cells, they do reproduce asexually. The main or most common way that they will reproduce is what we call binary fission. Snapping division and budding can also occur with some species, not necessarily all, but some of them. So binary fission, prokaryotes like to keep it nice, short, sweet, and simple. So you have in step 1, you would have a new cell. It's growing, it receives the messages, okay, let's get ready to replicate. It's going to replicate its DNA first. That has to occur before you can go through cell division, because each new cell needs its own copy of DNA. So as soon as that DNA is completed with replication, the cell will tend to elongate a little bit, separating those 2 molecules of DNA to opposite ends of a cell. Remember, prokaryotic DNA is circular, so it's easy to separate them. You start to get this pinching in, or what we call invagination of the cell wall. It's kind of, think of a balloon, and you start pinching it in the middle until it forms all the way across, and bingo, you now have 2 new cells. You don't have to worry about mitosis and the various steps, because prokaryotic cells don't do that. They go through this binary fission. By step 5, what happens to each of those 2 new daughter cells, which may separate, they may remain slightly attached, is then they will start through their own life cycle. How long does this process take of developing a brand new cell through the time when it will go and develop 2 new cells? So how long does it take? That's a generation time. It depends on the species you are talking about, and it depends on the environmental conditions. There are some species that we have studied for many years in the lab. We know everything about them, such as E. coli. Staphylococcus aureus is another one. With E. coli, it's been so well studied, we know what its optimum growth conditions are. So under those optimum conditions, from the time you get one brand new daughter cell to the time it will completely go through and produce 2 new cells, is 18 minutes. Think about that for a little bit. The next time you want to be grilling hamburgers and you choose to leave that hamburger meat sitting out, think about that. Every 18 minutes it's going through this. That's why, just cook your meat. Snapping, it's kind of a variation of binary fission. You basically form a new cell wall. The older one's going to literally kind of snap it because it bends a little bit, it ruptures. It's almost like you have a hinge there that you're moving. And then some will produce spores. This is showing a picture of what's known as actinomycetes. One thing you'll notice here that there are vegetative cells and then there are the spores. Actinomycetes are a unique group in that they form these filamentous vegetative cells that almost look like the hyphae on fungi. And indeed, actinomycetes are classified as bacteria, so they are prokaryotic. A lot of people feel they're almost like this link between the prokaryotes and the eukaryotes because they have some properties that are very similar to fungi. When you grow them in a liquid medium, they grow much more like fungi do. They will not turn the media turbid. Instead, you get these little balls, which is how fungi are going to grow in there. So that's why some people say, okay, genetically speaking, they are prokaryotes. But it's like they're this link between the bacteria and the fungi because they do have characteristics of both. For budding, what happens is the cell is growing and you kind of just form an extension off from that. As you can see, you form this young bud. It has its copy of the DNA streams into it, and then eventually that connection breaks between the original parental cell and the new daughter cell. There are some organisms that will have unique variations of the way they will reproduce. Some produce live offspring that emerge from essentially the body of a dead mother. So you have, in this picture, you've got several new cells still enclosed inside the original mother. They never separated from it. The arrangement of the prokaryotic cells, part of it has to do with, number one, keep in mind, we're looking at something just on one level, but there's multiple planes in which cells can divide, and do the daughter cells separate from the mother or not. In other words, just look at the arrangements. In addition to the morphology of whether it's cocci, and bacillus. And remember, in microbiology, yes, we're going to annoy you. We don't say circles and rods. We use cocci and bacillus. That would be the correct terminology. And keep in mind also that bacillus with a lowercase b refers to rod shape. Do not use the capital B, because that is going to refer to the genus Bacillus. I know, same word, but once again, I think we're here to annoy you sometimes. Just keep in mind those differences, please. So when you talk about morphology, you're talking about, is it cocci, is it bacillus, is it spiral? The arrangement is talking about what the community or what the group looks like. Are they in singles? Are they what we call diplococci? Just means that you have two of them together. Is it a chain? A long chain sometimes of them. Is it tetrad? Sometimes you will see cocci, especially in tetrads. So you have four of them connected together. Staphylococcus, that is where it looks like a bunch of grapes, that it's in multiplanes like that. And that typically is seen when you grow Staphylococcus from a broth culture, just so you know. You grow it on a solid medium, you don't always get that classic grape-like cluster. The arrangement of the bacilli can also be Diplococcus. It can also be in long chains. Palisades, where they line up instead of long end to long end, they're lined up in a row. Sometimes they're more V-shaped. In terms of classification of the organisms, there are three domains for all living organisms to fall into. That's the archaea, the bacteria, and the eukarya. The archaea and bacteria contain the prokaryotes. Eukarya contains the eukaryotic organisms. So this is one way of looking at the bacteria. As you can see, is larger than the archaeas. The archaeas were here from an evolutionary standpoint before the bacteria were. And they tend to survive in a harsher environment, high acid, oftentimes anaerobic conditions. Conditions where you might think, how does anything survive in this? Well, there's probably going to be some archaeas there. The bacteria, as you can see, you've got your gram-positive bacteria. You also have your gram-negative bacteria. If we look at the archaeas, some of the common features, they do have a cell wall, but it does not contain peptidoglycan. So that is one way you can distinguish between the archaeas and the bacteria. There's two main phylums. They do tend to reproduce by binary fission, budding, or fragmentation. They can be cocci, bacillus, spirals, or pleomorphic. From a medical standpoint, you're probably not going to be studying these because at this point in time, we don't know if any of that are pathogenic to humans. They tend to be studied a lot in environmental microbiology. Just to give you an idea, as I said, they are pleomorphic. This shows some pictures of some of them. And as I said, they're what would be classified as extremophiles because of the environment that they survive in. A lot of them are known to be thermophiles, being that they can survive in much higher temperatures than what you would expect. Hot springs. They can survive in halophiles, meaning they can survive in high salt concentrations. So the thermophiles, they are often surviving above 45 degrees Celsius. Just for reference, remember, human internal body temperature is 37 degrees C. Hypothermophiles, they require temperatures over 80 degrees C. So that is showing here some hypothermophiles that are living in hot springs, as I said. All that yellowish orange, those are the archaeas that are growing there. And then the halophiles have said extremely high salt concentrations, sometimes even greater than 9% sodium chloride. Some of these contain pigments that help to protect them from the sunlight. Some of these have been studied more than others. This is showing a picture of some halophiles that they have growing. The This is high salt water, and so they're using them to grow in that. That's what the coloration is from. They're trying to see if they can grow them in salt water and basically remove the salt from it and convert it to fresh water. Methanogens, this is still talking about archaeas. These are the largest group of the archaeas. They convert carbon dioxide, hydrogen gas, and organic acids to methane gas. So oftentimes what they're doing is they're converting waste products, organic waste products, to methane. Some of these will live in the colons of animals. I lived in a small town when I was in graduate school in northern Idaho, where it's a small enough community that their sewage plant, they actually use methanogens to break down the sewage. They're very effective in breaking it down, and what was the byproduct? It was methane gas. They then turned around and used that methane gas to heat their buildings on site. They also had a way of converting it to run all of their city vehicles that were associated with the sewage plant, ran off methane gas, and so they were totally self-sufficient from that standpoint. That's not necessarily feasible in a large community, but at the time this is a community of around 15,000, and so it worked for them. Some of your what we call deeply branching bacteria, these are usually autotrophic ones. They live in these habitats that are fairly harsh, that once again is felt to be similar to the way things were when Earth first started, when life first began on Earth. So the evolutionary standpoint, these are the first ones that were there. There are some phototrophic bacteria that are they're grouped based on the pigment that is present. They are capable of carrying out photosynthesis, so they're referred to as your blue-green bacteria, or their cyanobacteria, the green sulfur bacteria, green non-sulfur, purple sulfur, and purple non-sulfur. So like I say, these are classified according to the pigmentation that they have. So this is an example of the cyanobacteria. Like I say, they do carry out photosynthesis similar to the way plants would. So here's purple sulfur. This is a table just showing the different groups and some of the characteristics of those that are capable of carrying out the photosynthesis. So if we look at your bacteria, the one thing to start off with is they used to just casually kind of look at them, gram-positive, gram-negative. And now what they do is they further divide it into what they call the GC content, whether it's low or high. And this is referring to the percent guanine cytosine that's present in the DNA. So a low GC would have a low concentration of GC. It would be mostly 18. So if we look at the low GC gram-positive bacteria, we're going to look at just to go through some of these. Don't get bogged down by thinking about the diseases, because like I say, the diseases that are caused by some of these will be discussed in a later chapter. Clostridium is rod-shaped, so it's bacillus-shaped. They are obligate anaerobes, so they don't like oxygen. That's going to kill them. They're very important in both medicine and industry. There are some other organisms that are associated with them. Mycoplasms, these can be either facultative or obligate anaerobes. They do not have a cell wall. They're very small. Sometimes they will colonize the respiratory or the urinary tracts. They have a very classic look to them, which is referred to as a fried egg appearance. So when you grow them, it looks like you've got that fried egg and the yolk in the middle. That's going to be mycoplasm. There are some additional gram-positive that are low GC that are both bacilli and cocci. One that's bacillus is the genus Bacillus, very commonly found in the soil. As I mentioned a little bit earlier, Bacillus anthracis is the causative agent of anthrax. So that's of concern medically and also from a governmental standpoint. Bacillus thuringiensis is of interest in agriculture because it produces a natural toxin that is used by farmers now and gardeners as an insecticide. So this is showing the Bacillus thuringiensis, and that orange, that's a toxin. Listeria is, all of these are bacteria. Listeria is something that is oftentimes contaminating meat products, especially processed luncheon meats. It's been a problem. It can reproduce under refrigeration, so storage can be a problem with. Most people, it will not make you sick. The problem with this particular bacterium is that it can cross the placental barrier, so it can be a problem for women who are pregnant. Lactobacillus usually is not going to cause diseases in humans. It is used in the production of a lot of foods, especially what you would think of more like dairy-based foods, yogurt, things like that. Streptococcus and Enterococcus can both cause several different diseases. Streptococcus causes things like strep throat. Staphylococcus is going to be one of the most common things that you're going to find in humans. I can probably take a sample of anybody's skin and we're going to find Staphylococcus. You'd probably find it on any tabletop, doorknobs. It's just about everywhere. Can it cause diseases? Yes, it can. Usually the problem is it has to be able to penetrate your body in order to cause disease. It's contaminated food. This Staphylococcus is the one that in summertime can be a problem with potato salad. And then it likes your potato salad just like you do, and the problem is when you leave it out and it's not kept cool, Staphylococcus loves that. It starts growing and it can cause a type of food poisoning. All of the Streptococcus, Enterococcus, and Staphylococcus are all, as the names would imply, they are cocci, or spherical in shape. So this is just showing some of your gram-positive. By no means is this all the gram-positives. It's just showing you a selection of some of them. Certain characteristics associated with them, and in the far right it does show the different diseases that can be associated with them. Like I say, we will cover that later. Your high GC gram-positive bacteria, this will include things such as Corynebacterium, Mycobacterium. Mycobacterium typically grow very slow. They have a lot of mycolic acid in their cell walls. And then the actinomycetes that we saw earlier. Streptomyces is another genus. It's a type of actinomycete. Streptomyces do produce some antibiotics. They also produce things such as different compounds that have been produced from an industrial standpoint. I actually have worked with Streptomyces when I was working on my master's degree. The professor I worked with prior to my starting there is the one that worked with Streptomyces and determined the pathway of having them produce particular compounds, which he then sold the process to a company for making some of your artificial sweeteners like saccharin and things like that. So once again, this is showing with the actinomycetes the filamentous growth that they have. Gram-negative bacteria, this is going to be part of your largest of the groups. Very diverse group. This will include some bacteria that are very important for agriculture because they are nitrogen fixers. Aspergillium and Rhizobium grow in association with plant roots. Plants need nitrogen, but they cannot always use nitrogen in the form that it's found in the soil. And so what these bacteria will often do is convert it to a form that they can use. Sometimes these will have flagella on them. Some of them they'll have the procesa, which will help them stick to other surfaces. Rhizobium, they are known to form these nodules on your legumes, like your peas, and it's a nitrogen fixer. So it's converting nitrogen into a form then that the plant can use. Nitrobacter is another one that is important in the environment and agriculture. And some of these groups, it does contain, once again, some pathogens such as rickettsia and brucella. Both can cause human diseases. As we go through these different groups, it's mentioned some of the ones that do cause pathogens, but they're also mentioning some that have importance from an environmental or agricultural aspect. Campylobacter is kind of unique that it forms this unique shape that we saw earlier. Has a flagella on one end that helps it to move through the environment. It's sometimes found in aquatic systems. And the prosthetic, what it does is it allows it to stick to typically like rocks. And it allows it, if it's in a stream, the water's flowing really fast, this allows it to basically hang on tight. And it will hang on there as the water's flowing by instead of being washed downstream. And you'll notice it has a unique way of how it will reproduce. It doubles in size and then it snaps or splits, producing a new cell. And then with that flagellum, it's going to actually move over, attach to another rock, and start the process over. Sometimes you have swarming, where you have several of them come together and form almost what's known as a rosetta, kind of this star or rose type shape. Some of them can produce, as I said, diseases in plants. Sometimes they produce, bacteria will produce what's known as a plant gall, where you get this swelling. Typically it's right, you can have root galls, you can also have stem galls. If it's stem, it's right close to where the stem is emerging from the soil. Sometimes this is like an outgrowth. I think of it kind of like a tumor on the tree. And sometimes the plant can survive just fine with this on it. Sometimes it will extend and actually end up killing the tree. It just depends on the species and where the location is. In your beta Proteobacteria family, this is still the gram-negatives. This includes things like Neisseria, that can cause numerous diseases. Bordetella, which causes things like pertussis, that's whooping cough. Some of these will cause infections in mucous membranes. Some of them, it will be in, say, the respiratory passages deeper down, intestinal passages. It depends on the species as to, once again, we'll study later as to what disease it can cause. This is just giving you an idea of all these with the gram-negatives. Thiobacillus helps to recycle sulfur in the environment. Zoglia is used in treatment of sewage because it often forms what we call these flocks. It's going to help with the treatment of the sewage. And this is showing the flocks of how it helps. Basically, it's helping to prevent clogging up in the sewage treatment plants. Some of these groups you will have different subgroups. In this group, you have your purple sulfur bacteria. Once again, they can become pathogenic on other organisms, such as in the picture in B on a roundworm that it is affecting. Some become intracellular pathogens. Legionella causes Legionnaire's disease. This was diagnosed or was discovered, I believe it was in the 70s with the American Legion Convention, where several hundred of their members got sick at a convention. And they finally were able to determine that it was this bacteria causes kind of a pneumonia-like infection with the lungs. But it was in the air conditioning units, in the water. And so the water became contaminated that's used for cooling the air, and then through the air vents it's being blown everywhere in the facility they were at, and that's why so many people were getting sick. There are some of these bacteria that are methane oxidizers, so they'll use methane as a carbon source and as its energy source. In this case, they would be living in an anaerobic environment. Some of these are what we call glycolytic facultative anaerobes. They can break down carbohydrates by glycolysis, and then they divide into other pathways, multiple pathways that can be used. Now, there are three main families here: your Enterobacteriaceae, Vibrioaceae, and Pasteurellaaceae. Enterobacteriaceae contains a lot of bacteria that can become human pathogens. Things such as Escherichia, which most of you would be familiar with, Escherichia coli, or E. coli as we shorten it. Enterobacter, Serratia, Salmonella food poisoning, Shigella, which causes dysentery, Yersinia, which causes the plague, Klebsiella, which can cause pneumonia. So there are a lot of human pathogens that are in this group. Vibrioaceae, Vibrio causes cholera. So this is where a lot of your human pathogens are found within these groups. Pseudomonads, they are able to break down a lot of organic compounds, and so they're being looked at from environmental microbiology in terms of bioremediation, using microorganisms to break down hazardous materials. Pseudomonas can also be human pathogens. They can cause a lot of urinary tract infections, ear infections, lung infections, and they're also commonly found in the soil. And one of the problems now that we're seeing is increased drug resistance. Pseudomonas typically has flagella, and so if you do a flagella stain, you can often see some pretty cool pictures where some of them have multiple polar flagella where it's on the end and there's multiple flagella from one side. So they're kind of cool to see. Additionally, they're still continuing with bacteria, things such as Desulfovibrio or Delavibrio. Delavibrio is interesting because it is a very small bacterium. Actually, it is a pathogen of other bacteria. So that's a little different. And Myxobacteriums are very small. They initially were thought to be like slime molds. So you have your vegetative cells that are going to grow. You get a whole mound of this. It starts forming this mound when you're depleting the nutrients. And then they form this fruiting body with the spores on the end that then are released. Each spore then can travel and germinate into a fruiting cell, and it starts this whole process again. In this last group, we have Campylobacter and Helicobacter. And then additional family where we have the Proteobacteria. Only 2 organisms have been able to be cultured. They're commonly found in the ocean. It's hard to culture bacteria or anything from the ocean because, especially as you go down deeper in depth, the pressure increases. And so a lot of those organisms, when you try to bring them to the surface, they essentially pop because of the change of pressure. You're also dealing with, out in the ocean, salt water, so that means they're used to a higher salt concentration. You have to try to match that. There's just a lot of variables that make it very difficult to try to grow them. Chlamydias tend to grow intracellular in both animals, birds, and some invertebrates. It's the most commonly sexually transmitted bacteria in the United States at this time. Spirochetes, these are motile bacteria. They move in this corkscrew type motion. They have very diverse habitats they can live in. The 2 that can cause diseases in humans are your Treponema and your Borrelia. There are bacteroids, which some of those can also be human pathogens. They can affect digestive tract. They can also be pathogen on other animals.